We're very pleased today to have Dr. Fahd Qadir here with us from Tulane. His talk is titled From Sex to Systems, Molecular Signaling in Beta Cells in the Path to Precision Diabetes. Just a little bit about him. He's a computational and systems biologist investigating how steroid receptor signaling and sex chromosome biology influence pancreatic islet cell function with a focus on mechanisms driving the chronic metabolic disease epidemic. He's a postdoctoral fellow at Tulane University School of Medicine, mentored by Dr. Frank Mova Jarvis, and a recipient of the NIH NIDDK K99R00 Pathway to Independence Award. Congratulations on that. That's very difficult to get. He also completed his PhD in molecular cell and developmental biology at the University of Miami as a Fulbright fellow underneath the mentorship of Dr. Juan Dominguez-Bandela. I'm going to drop a couple of his papers, this new one and a previous one where he shared the lineage of the beta cells in vitro. And thank you so much for joining us. If you have a question, drop it into the chat and he can take questions during or at the end, whatever you prefer. Take it away.
All right. So thank you, Monica, for the invitation. It's second time I've been here. First time was for one of my PhD papers with Juan. So it's always nice to be here in sugar science. So I'm going to be talking a little bit about some of the papers that we've actually published and the work that's ongoing in our lab. Monica already told you a lot about me, but I'm sort of an islet and developmental biologist who transitioned to computational and systems biology purely because all of the interests that I've worked under have begun to understand that, diabetes and overall metabolic health is systems biology problem, and it's not restricted to one particular cell type or organ. So I'm also a recipient of the K99R00, which was activated quite recently. So I'm going to be talking about some of the work that we're doing. So I have nothing to disclose. I'm a poor postdoc. So I'm going to start off by putting out one of these observations that my mentor actually, because being a clinician, he brings this unique perspective into studying these systems-wide developmental diseases or metabolic diseases. And one of the things that he always has been telling me about is that type 1 diabetes is one of the unique autoimmune diseases that does not have the female predominance that you see across all other autoimmune diseases. So it's an open question in biology that why is this? What is the reason that males have a higher predisposition to type 1 diabetes than females do? So what are the ways in which sex differences can occur in terms of systems biology? Well, there are two different ways in which differences can happen in terms of sex. One is via the sex chromosomes. Of course, males have the X and Y chromosome, and females, they have two X chromosomes, but one of the X chromosomes is silenced by this long non-coding RNA known as the XIST, or the X inactivating transcript, which is basically a very unique and very interesting long non-coding RNA because it literally wraps all around this X chromosome, preventing gene expression. Anyway, so one of the differences, the way in which differences can occur is via sex chromosome, but the other way is due to these steroid sex hormones. So the predominant hormone in men is testosterone, and testosterone also occurs as an analog, which is more potent. And by higher potency, I mean that its binding affinity to its receptor, the androgen receptor, is about 2 to 3 times. And some people say it's 10 times higher. In females, the predominant hormone is estradiol 17 beta, and during pregnancy, you have higher progesterone. So the varying concentration or ratios of these hormones across different sex also exert sex-specific differences. So that's why either pubertal hormones or sex chromosomes in terms of gene expression can exert differences. So we've always thought of testosterone, us in the lab, because we study this all the time, but people think of testosterone or estrogen, or estradiol 17 beta specifically, being either a female or a male hormone. Well, that's not entirely true, because males, although they make a lot more testosterone, they also make a certain amount of estrogen. And the majority of estrogen actually comes from testosterone via the interconversion, via the enzyme aromatase. In females, the majority comes from the ovaries. Again, it's the conversion of A4 androstenedione to testosterone. And the majority of the estrogen, of course, is coming from the ovaries. In postmenopausal females, a lot of the ovary production of E2 shuts down. So a majority is then coming from A4 rather than being directly produced in terms of estrogen. But what's interesting is really Frank recently published a paper with a collaborator of his, and they looked at what are the ratios in production of these steroid hormones over life. And what was striking to me, at least, was that if you look at female testosterone, you'll see that it's actually way higher in terms of picograms per ml. This is very reliable data because it's taken from the NHANES data set of the NIH, which is where they actually detect these hormones via mass spec, which is the most reliable way of checking these steroid hormones. So you can see it's about, at its peak, round about, just below 400. And estradiol 17 beta at its peak is, somewhere in the 100 range. So about, 3.5 to 4 times more testosterone in females than estrogen. Of course, males have a lot more, a log difference more, but the evolving concept that we have now is that, it's more about the ratio of these hormones. But what's important that I'm trying to point out over here is that both of these hormones across the life of males and females are present. It's just that the ratio in which they exist differs. So now coming to the second big difference is the X chromosome and the Y chromosome. The X chromosome is like this very interesting long stretch of active and inactive sites of various genes that either control how the overall genomic environment of all chromosomes is regulated, because they have a lot of different KDMs or demethylases that exist on them. So just to study the overall structure, there's this large XAR region, which is known as the X added region, which was added to this chromosome just after life decided that the placenta was important, or just after Eutherian evolution. And then there's this small region, which is about only 850 kilobases in humans, known as the X inactivating center. And this is where all the magic happens. This is where the XISD actually is engaging via other different proteins that bind, forming the X inactivation complex, which then inactivates this entire chromosome. And, in a series of experiments that others have done, not us, that, if you take this X inactivating center and if you put it in an autosome, you can actually silence an autosome. So this is a really important region when it comes to the silencing of one X chromosome in females. Then there's the XCR, which is a conserved region of the total X chromosome. There are these genes, the androgen receptor, interestingly, is actually present on the X chromosome. XIST, and then its negative regulator T6 is present over here. These two chromosomes, they basically orchestrate, sex differences, because different sets of genes, they exist across these, two different chromosomes. Okay, so my mentor is basically a leader in the study of all of these receptors and sex differences that occurs in terms of overall the systems biology of metabolic diseases. So he's done a lot of hallmark work in his lab, looking at how, estrogen receptor alpha, for example, can promote cellular survivability in terms of beta cells, which is sort of the focus of some of the work that I do, right? So if you look at ER alpha, for example, he's shown that ER alpha signaling actually prevents cell death, and it promotes, cellular resilience in beta cells. What the exact mechanism is, how this is happening, that is, something that is off a study in our lab, and it's not something that, we know really well, but ER alpha signaling is important both in male and female beta cells. So then I'm going to talk about testosterone. It's a majority of this talk because that's the paper that we recently published. So one of the largest randomized placebo-controlled trials, which was actually done in Australia, about 1,000 men. And what they studied was, can testosterone actually prevent or delay the onset of type 2 diabetes? And in those 1,000 men, they found that, it actually decreased the abnormal OGTT in about 41% of individuals when compared to the placebo. So it seems like testosterone is really important in terms of overall metabolic health and health. So the question is, how do these hormones actually, how are they influenced by how are beta cells, or how are different metabolic tissues affected by these plasma steroid hormones? And as I said, there are a lot of other players in which are also responsible, for example, the conversion of testosterone to DHT. And one of those enzymes is SRD5A1, or 5-alpha reductase, which converts testosterone to DHT. And another important aspect is when you're studying, specific tissues and their action is that, do beta cells, which are the cells of our study, do they actually contain the receptor for these ligands, which is the androgen receptor? So then from a systems biology perspective, how does testosterone actually interact with the brain, with all of these different tissues, the gut, islets, how does this all come into play? And we're studying all of these tissues and these interactions one by one. So how does androgen receptor signaling work? So testosterone in the plasma is actually bound, the majority of it is bound to sex hormone binding globulin, about 60 to 65%. And the majority of what's left, about 38%, is bound to albumin. What's left is really 1 to about 3%. That is like free testosterone. The majority of that testosterone is what is actually exercising these actions of testosterone in different cells. So testosterone is a non-polar steroid, so it doesn't need a canonical receptor to traverse these plasma membranes. So because it just dissolves across the lipid rafts. Once inside cells, it can be converted into DHT or 5-alpha reductase, and it's literally just reducing the C4 to C5 bond over here and adding a hydrogen. So the structure of DHT and testosterone is very similar, but this small reduction actually makes this much more rigid and increases its affinity in terms of binding to the androgen receptor. Androgen receptor exists inside cells bound to these heat shock proteins, and once DHT binds, it forms this dimers, heterodimer of androgen, which then can act on the genome by traversing the membrane, binding, forming this complex, and then driving gene expression. That is a genomic one. So another non-classical mechanism by which DHT can act, depending on the concentration of either testosterone or DHT, AR can exist in a monomeric form, and then it exerts a different function, or it goes through these cytoplasmic actions within beta cells or within other AR or testosterone or DHT responsive tissues. So this is a really interesting paper, and again, something that Frank wrote a couple of years back. When I joined the lab and I started looking or studying, differences in terms of steroids and sex and why this is so important when we're looking at precision therapies catered to men or women, he told me you need to read this paper first. So when I went over this paper, it really helps orient how you're going to go about performing these studies, because they can be incredibly complex. Because as I giving you a small sort of overview, there are a lot of different players which come into play. So first thing you want to see, is there an observable trait? Is there a sex bias trait? Some causative disease, which is more prevalent in either males or in females. If there's none, then it's just a similar biological effect, which is same across all sex. But if that does exist, then the first thing you need to do is to test for pubertal hormones, gonadal hormones after puberty. And if that is true, then you need to identify the receptor by which that action is happening. If, again, you reach no after testing for those gonadal hormones, and you need to check for perinatal masculinization. So in utero, in males, there's a surge of testosterone that happens that masculinizes the brain, reorients the genome, and that remains persistent throughout life. And it has a similar effect across all different, metabolically responsive male tissues. So it's important to study these tissues, because this is also one of the effects of the sex hormones. So at that point, you want to investigate, is testosterone acting by AR? Is it being aromatized to E2? What is the mechanism of its action over there? If you still reach a no, then it's a cell autonomous effect due to gene expression differences across cells. And then, of course, you want to start identifying what those genes are. And if you're not seeing something here, then there was something wrong in the way that you're observing these differences. So Frank, again, a couple of years back, published this really hallmark paper looking at how androgen receptor action via DHT actually increases glucose-stimulated insulin secretion, or GSIS, in male mice. So what you're seeing over here is the islets isolated from these control mice to just pre-mice, and you can see when you add DHT, there is an increase in GSIS, which can be quantified over here. In the right-hand panel, you can see that when you actually knock out the androgen receptor in a beta cell specific way, you will actually lose that responsiveness, and that's what you're seeing over here in the total AUC. But when you compare the DHT treated in the control mice to the knockout, you'll see a very profound difference. So there's even a difference that exists, honestly, between just the knockout to the control. So natively, in islets that are not exposed to DHT, but don't have the androgen receptor, there's reduced GSI. So obviously, androgen receptor in male mice at least is important. So then the next question is that do beta cells express the mechanism for the interconversion of testosterone into DHT? And in a paper that we published again a couple of years back, we showed that that's true. So the enzyme SRD5A1 is the predominant enzyme expressed in beta cells and in bulk islets in terms of gene expression. And also in terms of looking at the protein expression, you can see both in males and females, SRD5A1 is expressed, and the androgen receptor, which is the receptor to which testosterone binds, is also expressed in beta cells. So what is the story of how androgen receptor, because after the cell metabolism paper, it became clear that it's somehow working with GLP-1, but we didn't really understand how this was happening. So in a series of very elegant experiments done by one of my co-postdocs, when she was in the lab, Dr. Paula Motoresa, what she did was that she did an IP pull down of androgen receptor from beta cells in Th1nk, in insuloma cell line as fitted from rats, right? So when she did that, she found something very interesting. That she found that AR actually binds to a plethora of different proteins. Then, some are responsible for mitochondrial function, others are kinases, phosphatases, some are responsible for insulin vesicle maturation. The actin cytoskeleton was another important one. All of these are important in, overall beta cell health. But what we found the most interesting is that they also bound to a lot of G alpha subunits or GPCR subunits specific to the GLP-1 receptor. So it seems like AR was binding to this and exerting some, helper function to GLP-1R and increasing its activity. So the question is, how is this actually happening? So the first thing we did is this experiment that I did, we performed single-cell RNA sequencing, and this is just a UMAP showing all of the different cell types that we were able to identify. This is just some quality control on the right. And what we found was very interesting, but we found a majority of the genes that were downregulated in so these are two different types of beta cells based on the expression of insulin. This is being described in literature before. And we found that, a majority of the genes that were downregulated were actually mitochondrial genes which are, transcribed by the mitochondrial genome or associated with the electron transport chain. So it seemed like a lot of pathways responsible for electron transport or oxidative phosphorylation were actually downregulated in these beta cells. So then we went, I'm not going to show you the wealth of that data, but in the interest of time, but we went about performing a series of experiments, which included mass spec, looking at metabolites, and also this other specialized protein acid known as the RPPA, reverse phase phosphoproteomics, and looking at gene expression, combine that with gene expression as well. So we had like this composite picture of what is happening in terms of metabolism inside these beta cells. And all of these genes, which are seeing all these enzymes, which you're seeing in blue, are actually upregulated. That we found them using different types of tests, either transcription or protein expression, or by looking at metabolomics. So it seems like there is an increased turnover of glycolysis and the TCA cycle, but a majority of genes which are responsible for the ETC or the electron transport chain are actually downregulated. So it seems like there's a shift from oxidative phosphorylation to glycolysis and TCA. Now, this is interesting because what we found, one of the genes that were upregulated were pyruvate dehydrogenase. What it does is that it converts pyruvate into acetyl-CoA, and in doing so, it generates a lot of carbon dioxide. And this is important because this increased carbon dioxide is then converted into bicarbonate via the enzyme carbonic anhydrase. And this bicarbonate actually is a substrate for another enzyme, soluble adenylate cyclase, which is a evolutionarily conserved bicarbonate sensor. And that's what converts ATP into cyclic AMP, increasing cyclic AMP tone inside a beta cell, allowing a beta cell to perform its action of insulin secretion in a glucose responsive manner. So it's a bit complicated, but what I'm trying to show you over here is that DHT seems to be increasing this carbon dioxide load. We're going to be talking about that in the next slide. So we actually did this experiment, and again, we did another series of experiments, but I'm only showing you the one which is the most important over here. And what we found was, in our overall pathway, you have increased turnover of glycolysis in the TCA cycle. So you have a lot of this carbon dioxide that's produced, and it's converted into bicarbonates via this enzyme carbonic anhydrase. So, I want to tell a little story about carbonic anhydrase because I think they're very important enzymes. And if you think of life in general, there is this very important enzyme, which is the most predominant protein on the planet, known as ribisco, ribulose 1,5-bisphosphate carboxylase or oxygenase. And this enzyme is present in plants, and what it does is that it actually fixes carbon dioxide and it adds it to ribulose 1,5-phosphate, converting it into 3-PGA. And this is a very important enzyme, if you remember from high school biology, as part of photosynthesis. But what people don't tell you is that ribisco is actually a very inefficient enzyme. And it depends on whether enough carbon dioxide or enough oxygen is present. And based on that, it can shift from either doing photorespiration or photosynthesis. The enzyme which is responsible in plants for causing the accumulation of carbon dioxide is actually carbonic anhydrase, because it makes this sort of reversible balance between the amount of bicarbonates or carbon dioxide that is present in plants. Now, the reason I'm telling you this story is that the analog, alpha carbonic anhydrase, which is present in plants, is also present in humans. So that's the one sort of similarity that plants and humans have, is that we share the same analog. We have about, I think, 7 different isoforms of carbonic anhydrase in humans, but we have alpha carbonic anhydrase, which is the same, which is present in plants. Now, if you think of carbonic anhydrase, what is doing over here is that it's taking this carbon dioxide. And this is super important, because if you have accumulation of carbon via carbon dioxide inside cells, carbon dioxide is very inefficient in being transported, very poorly soluble in water, unless it converts into bicarbonates. And carbonic anhydrase increases makes that process faster. And that's how cells are quickly able to remove all of this carbon dioxide that's been produced from respiration. So we didn't see a difference per se in carbonic anhydrase because it's an homeostatic enzyme. But what's happening over here is that cells, because they're producing now much more carbon dioxide because of glycolysis in the TCA cycle, there is an increased action of this carbonic anhydrase converting it to HCO3. And that's what I was telling you over here, where it actually acts as a sensor. This adenylate cyclase is actually a sensor for bicarbonates, and it converts ATP to cyclic AMP, which results in insulin secretion. So we actually did experiments that if you block carbonic anhydrase, what happens to GSIS in a DHT specific manner? And we found that there was a decrease. So you can actually see when you had DHT versus DHT and carbonic anhydrase inhibitor acetazolamide, we found a trend showing a decrease in overall GSIS and human ions. Similarly, if you were to inhibit the adenylate cyclase enzyme using adenylate cyclase inhibitors, LRE1 or TDI-10, 2 to 9, you can again see a decrease in DHT's insulin shrink action in beta cells. So this is our overall hypothesis of what is going on inside a beta cell when exposed to testosterone. So what testosterone does is that it either remains as testosterone is converted to DHT, binds to androgen receptor. Androgen receptor then has this action within mitochondria, where it increases the action of PDH and IDH, increasing carbon dioxide, which is then converted into HCO3. AR also goes and binds to the nuclear genome, which causes a difference, in terms of overall mRNA gene expression, and specifically glycolytic genes. And AR also sort of is recruited to the GLP-1 receptor, perhaps stabilizing it, or this is something that we need to investigate more, but we know that it interacts with GLP-1R, perhaps stabilizing it, allowing for the action of soluble adenylate cyclase to be amplified. And it also is, HCO3 is very important because, as I said, that's how, cells shuttle carbon dioxide out of the cell. And we also found when GLP-1R is internalized in different microdomains within the beta cell, we also found cyclic AMP and AR action to be present over there. So it increases cyclic AMP at the endosomes, and increased cyclic AMP is involved in insulin granular exocytosis. So this paper all of this, what I'm showing you, is actually published in this paper and Cell Reports. So now I'm going to move to the second arm of my talk, which is more on sex chromosomes. So like looking at gene expression differences. So one of the big questions we had is that in the absence of hormones, what are the differences that drive males and females in terms of gene expression? And we have a unique system in terms of, all islet polyps, not us, but when you actually isolate islets from human donors, they're no longer in the presence of steroid hormones, right? Because the dish doesn't have steroid hormones unless you add it. So this, sort of affords us the unique opportunity to study what is the action of gene-specific differences, which in majority exist due to sex chromosomes. So we did this experiment where we isolated islets, and then we did a series of different experiments from both males and females. We did a series of different experiments looking at the accessible genome, the transcriptome, and we did a series of experiments like looking at bioenergetics and hormone secretion. I'm not going to show you any of that data in the interest of time, but it's all published anyway. But so if you're interested, you can go and look at our paper. But I'm going to show you some of the ATAC-seq and the RNA-seq data. We also added a lot of donors from the HPAP dataset, which is a wonderful dataset, organized by the Human Pancreas Analysis Program at the HIRN. So this is as some quality control. I'm going to breeze through this, but it showed that our cells were sequenced optimally, and all of the sequence parameters, including total genes that were detected and total amount of genes that were successfully mapped to the HG38 genome, we all had high percentages for everything. So it seems like our data set was optimal. We ended up with 141,000 cells, single cells, and like these are all the different clusters. We didn't see a cell type specific or sex specific skewing of any of the data. So we found an even distribution in terms of sex or origin across all of our samples. And this is our ATAC data set, about 50,000 cells. And there was a strong correlation, and that's what you're seeing over here, between the expression in terms of RNA of a cell type and its subsequent accessibility in terms of the genome. So we wanted to start performing differential gene expression testing. Now, classically, across single-cell RNA-seq studies, people use a cell-by-cell strategy when performing statistics. And that's okay when you have limited power by about, let's say you have an N of 3 in your experiment. But in our situation, we are afforded by having, very high power. We had over 50 individuals. So we went about like trying to say, okay, how can we be a little bit more conservative in our approach to find something that is actually more applicable to a population rather than just looking at a subset of individuals. And what's interesting is, and this is something that I would like to bring to your attention, really, when looking at any type of single cell data set, you will see a lot of diversity in the number of cells. So this is just a plot showing you across all of the 52 donors, the overall distribution of cells. So if you were to use a cell-by-cell statistical method, you would have a lot of skewing of your data to those individuals which have a lot of cells. So for example, if you're looking at my pointer, I'll show you like this blue line is beta cells. And you'll see these two donors have a lot of beta cells. And we found that they're roughly about 5% of all beta cells and amongst all donors. So if you're doing a cell-by-cell basis, your statistical data is going to be just skewed towards these two individuals. So in order to prevent that, what we did is that we performed a pseudo-bulk approach where we compressed all of the reads of what you're seeing in all these beta cells and normalize them to 1. So it wasn't just a pure compression. It was a sort of normalized aggregate approach that we did. And then we performed a donor-wide comparison. So every donor over here is an N equals to 1 rather than every cell. So you can see that, again, we have cell type specific gene expression across all cell types, both in terms of the pathways and in terms of specific genes across each of our subtypes. Now, when looking at sex differences, we found that predominantly the biggest differences that we could see were restricted to sex chromosome genes, both in terms of X chromosome genes in females and Y chromosome genes in terms of males. And if you look at the pathways, it seems like a majority of the pathways that we found were linked to demethylation of the genome. And that's because in males, you have these Y chromosome specific demethylases like KDM5D. And in females, you have these X chromosome specific demethylases called KDM6A. Now your question will be, well, in males and females, you both only have theoretically one active X chromosome. So why is it that you have a higher expression of KDM6A in females? Well, there are some genes on the X chromosome that escape an X inactivation. And KDM6A is one of those genes. So even though the X chromosome is inactive, there are some genes that escape that inactivation and are expressed in the biallelic manner. And KDM6A is one of them. So it has a heightened expression in females compared to males. And we think that these are important because they may pattern the genome. And others have shown that these demethylases have a very essential function in how they pattern the genome in males and females. So we can actually correlate gene accessibility to the expression in RNA expression. So everything goes hand in hand, and in terms of the accessibility of these genes that I'm showing you, and in terms of RNA expression across different cell types. So the next thing we wanted to do is we wanted to be a bit ambitious and try and understand if we could link the accessibility of particular genes or the accessibility of motifs of particular transcription factors and the activation or expression of these particular genes. And we found this very, very cool tool that was published by Fleck and other co-authors in Nature, where they were studying neurons, and they essentially made this machine learning algorithm called Pando. And what Pando does is that it says that for every gene that's been expressed, you can create a mathematical function that looks at the accessibility of all the different transcription factor motifs that lie upstream to that particular gene, or which are distal to that particular gene. I'm not going to say this upstream, could be downstream as well, and we have distal tumors as well. So the mathematical function can be designed in such a way that you look at the accessibility of these motifs, and then you look at the expression of the transcription factors that bind to this particular motifs, and you can link all of this together to the expression of that gene. And it's an elaborate mathematical formula, which is depicted over here, but they do this via this machine learning module known as XGBoost. So it makes a relationship, a nonlinear relationship, between the accessibility of a transcription factor, the transcription factor motifs, and the expression of the transcription factors and the expression of a gene. And you end up with a map that looks something like this, which can be a bit overwhelming to study, but what I've painted over here in red are beta cell specific transcription factors. So you can see that there are differences in the overall activation scores when looking at a male gene regulatory network to a female gene regulatory network. And doing studies like this is important because this can really help us understand how gene programs are different between different sets of cells. And our experimental paradigm involves us studying males and female beta cells, but this approach can be applied to any question that you may have. So we also had type 2. We afforded this privilege because we got data from the Human Pancreas Analysis Program, and we looked at differences in type 2 diabetes. And we found this resounding difference in males and females, where we found about 1,800 dysregulated genes when you look at beta cells of females compared to about 210 differentially regulated genes in males. So what this told us, or what we think that this is telling us, is that a lot more needs to go wrong with a male beta cell in comparison to a female beta cell before it will become a dysfunctional type 2 diabetic phenotype. So female beta cells, ergo, female beta cells are way more resilient to the onset of some sort of pathology in terms of type 2 diabetes, at least, as compared to males, because a lot of the genes that we found dysregulated were related to mitochondrial function, not really to insulin secretion or insulin production. But in males, the predominant number of genes were involved with the insulin secretion and insulin receptor action in beta cells. So that just says that female beta cells in itself are just way more resilient, where you need about 10 times more genes, roughly 10 times more genes need to be dysregulated before you can get a type 2 diabetic phenotype in females. And again, when you look at the overrepresentation analysis, the pathway analysis of all these genes, you'll see, again, peptide hormone secretion is all downregulated in males. Basically, what I was telling you right now, but OXPHOS is downregulated in females, and we see a similar pattern in alpha cells as well. So now, in collaboration with Dr. Kyle Galton's lab, and this is work done by a very smart graduate student in his lab, Dr. Ruth Gamalna. She's graduated and moved on to bigger things. But what she did was that she took our data and she performed a cell type specific sex stratified GWAS, looking at the Th1nk data set, where she classified the data set into males and females and looked at SNPs or credible sets across these two data sets, looking at taking our accessibility profiles and looking at how these SNPs may originate or, really sort of be associated with the onset of type 2 diabetes in a cell type and a sex-specific manner. So we found interesting a lot of different hits, which are either sex-specific or cell type specific, across different cells. So for example, glucokinase, we found that to be present in, particular cell types. And for example, CCND1, which is a gene responsible for cycling. In gamma cells, we found that. So, in delta cells, we also found KCNQ1, which is a potassium channel subunit, I believe. So we found a lot of interesting hits. So this is just one example of, how cell type specific variants can exist and how they can influence disease. So this data is published, just to give you a wide open summary of the major differences that we saw in type 2 diabetes were oxidative, damages or decrease in oxidative phosphorylation in female beta cells, but in male beta cells, the differences are related to insulin secretion. And big differences that we found in terms of gene expression were these two candidates, KDM6A and KDM5D. And all of our data is actually uploaded to GEO and the Common Metabolic Disease Genome Atlas. So if you're interested in asking a question, we have all of these data sets available online. So you can actually go to our browsers, which are hosted on the CMDGA, and I have the links over here below. You can, literally go from hypothesis to publication grade images in 110 minutes, depending on what kind of question you have, both the accessibility information in terms of the ATAC-seq or the RNA-seq data set is present over there. So my last slide, most important slide, is acknowledgements. I really like to thank my mentor, Frank, for giving me the opportunity to ask all of these very interesting and cutting-edge questions, and providing the environment and the lab at Tulane really for me to grow as a scientist and allow me to actually apply for this very prestigious award, which I'm very grateful to have. And also a lot of other people in the lab who have done a lot of the work, especially our collaborators at UCSD, at Tulane, and the University of Miami as well. And most importantly, we'd like to thank the islet organ donors and their families. Without none of this work would have been possible. We'd like to thank the Th1nk, of course. I'm funded by the K99. I have a couple of awards also, a presentation coming up at the ADA, which I'm going to be talking about the work that I'm doing as a part of the K99. And Frank has a couple of funded grants from the Th1nk, from the NIGMS, and from the VA. So, follow me on Twitter if you have any questions. I'll take them now. Thank you very much.
Thank you, Fahd. That was a great talk. I had a quick question. Obviously this paper is focusing on type 2, but what can you say? I mean, there's been some work prior to this, and I even included that paper in the chat as part of related papers. There's been some work on type 1. What can you say from these findings, or have you kind of extended it into the T1 realm specifically at all?
No, we haven't actually looked at the type 1 realm specifically. Actually, we wanted to as a part of the ENBO paper, but the journal editors actually started saying that your paper is already very overwhelming as it is. So perhaps look at that independently. But yes, I mean, it's an open question. As I said, we don't really know. Kyle has been doing some really nice work in his lab looking at differences in type 1, but I don't think anybody has really done any sort of sex-specific work. So yes, that's definitely something that we're working on, but we just don't know really what is the major differences that occur in terms of sex.
Yeah, that's right. I mean, there's another paper that came out, the molecular mechanism for sex differences in electrical activity of mouse pancreatic beta cells. And again, they're looking really kind of at type 2. So and that was Petronal, Th1nk and company. So yeah, it would be great to kind of get at some of these questions, because right now there's a lot of buzz around, the essential question, is it the beta cell itself? Is it murder or suicide? I mean, and there's some great work coming out of Sarah Richardson's lab that showing that some of these endocrine objects in type 1 diabetics are actually smaller. So then what does that say about, some of the things you're presenting here? Really interesting.
Well, what's interesting is that, we think of, well, at least I think of steroid receptors as part of these homeostatic mechanisms inside cells, where they're working like little organizers, if you think, they're like picking up proteins, taking them where they need to be, acting like chaperones in a way, although they bind to chaperones themselves. But ER alpha, for example, and this is work that Fahd has already shown in his lab, that ER alpha actually promotes cellular survivability in terms of beta cells. So that automatically, by modulating how beta cells handle misfolded proteins, like the unfolded protein response in beta cells, it actually increases that, and that's one of the mechanisms how ER alpha signaling in female beta cells makes them more resilient. So that in itself, sort of will make beta cells more resilient in terms of any sort of, external or internal damage, be it from an autoimmune attack or from some sort of metabolic damage that's happening. So it's very important. Another thing is that everything that we study inside a dish, we don't actually recapitulate the physiological environment that happens inside the body. So inside the body, you're always exposed to different varying amounts of these ratio of these steroid hormones, and they have a different effect. And we've shown that in a dish, if you take iris and you expose them to testosterone or estradiol 17 beta versus not, they'll give you a different secretion profile, So, I mean, there are open questions we don't really know.
Yeah, I mean, if you want to extend that even further, it would be really interesting to look at some of the effects of these endocrine disruptors that we have in the society today, in our world today, and
Yes,
how they are impacting this system as well.
exactly.
Yeah, so it's really interesting. Congrats again on your award and of this really, really meticulous work. It's really exciting, and we'll look forward to seeing you at ADA and hopefully to talk again.
Yes, absolutely. Thank you for the invitation.
Thank you. Thanks again for coming, and we'll talk again soon. Thanks, Fahd.
All right. Thank you.
Bye.